Dr. Thomas Seyfried and other proponents of the metabolic theory of cancer argue that cancer cannot fundamentally be a genetic disease due to several key observations and scientific findings that challenge the genetic mutation hypothesis. Here is a detailed breakdown of the reasons behind this perspective. 1. Cancer cells' dependence on fermentation, not genetics. Cancer cells, unlike healthy cells, rely heavily on a process called fermentation to generate energy. In healthy cells, energy primarily comes from oxidative phosphorylation, a process in the mitochondria that uses oxygen to produce ATP energy. Cancer cells, however, exhibit a phenomenon known as the Warburg effect, where they generate energy through fermentation even in the presence of oxygen, a less efficient process that produces lactic acid. This is typically a backup energy process used in low oxygen conditions. Dr. Seyfried argues that this switch to fermentation is not driven by genetic mutations, but by damaged mitochondria that lose the capacity to undergo oxidative phosphorylation. This damage is observed across nearly all cancers and points to metabolic dysfunction rather than genetic mutations as the primary cause of cancer. 2. Mitochondrial damage and its central role. In healthy cells, mitochondria control cell growth and death, apoptosis. When mitochondria are damaged, they fail to regulate these processes effectively, leading to uncontrolled cell proliferation, a hallmark of cancer. Studies show that when mitochondria from a cancer cell are transferred into a healthy cell with a normal nucleus, the healthy cell often exhibits cancerous traits. Conversely, when a healthy nucleus is transferred into a cancerous cell with damaged mitochondria, the cell continues to exhibit cancerous behavior. This implies that the dysfunction in cancer lies primarily in the mitochondria, not the nucleus. This mitochondrial-centric approach counters the genetic mutation theory, suggesting that the origin of cancer is in the cell's energy system rather than in DNA mutations. 3. Mutations, a downstream effect, not the cause. Dr. Seyfried's theory posits that the genetic mutations observed in cancer cells are a consequence, not a cause, of metabolic dysfunction. When mitochondria are damaged and cells switch to fermentation, reactive oxygen species, ROS, levels increase. ROS can damage DNA, leading to mutations. Thus, mutations seen in the nuclear DNA of cancer cells may result from the oxidative stress created by dysfunctional mitochondria rather than being the initial cause of cancer. This perspective is supported by the presence of numerous passenger mutations in cancer cells that do not drive cancer progression. 4. Genetic variability within tumors. Cancer tumors often exhibit extreme genetic variability, with different mutations occurring in different parts of the same tumor. If cancer were solely caused by specific driver mutations, this diversity would make treatment virtually impossible, as there would be no consistent target across all cancer cells. However, despite this genetic variability, almost all cancer cells share a common metabolic characteristic: reliance on fermentation. This points to a unifying feature that is metabolic rather than genetic. 5. Cancer in animals and non-human primates. Studies indicate that cancer rates are much lower in wild animals and indigenous human populations that live without modern lifestyles. However, when these groups are exposed to processed diets, pollution, and sedentary behaviors, cancer incidence rises. This suggests that environmental and lifestyle factors that affect cellular metabolism, such as diet, toxins, and lack of physical activity, play a more significant role in cancer development than genetic mutations. 6. Cancer cells can grow without driver mutations. Some cancers have been shown to grow aggressively, even without the presence of driver mutations, which are thought to promote cancerous behavior. Additionally, normal cells can carry many of these so-called driver mutations without developing into cancer. If genetic mutations alone caused cancer, then all cells with these driver mutations would invariably become cancerous, which is not the case. This observation further weakens the genetic mutation theory and supports the idea that metabolic dysfunction plays a primary role. Summing it up, the metabolic theory as a holistic explanation. According to Seyfried, the metabolic theory provides a comprehensive framework for understanding cancer. Cancer cells' reliance on glucose and glutamine for fermentation-based energy supports the idea that targeting cancer's metabolic dependencies, e.g. via ketogenic diets, fasting, and mitochondrial supportive therapies, could be an effective way to manage and prevent the disease. By viewing cancer as a mitochondrial metabolic disease, it reframes prevention and treatment strategies towards supporting metabolic health rather than exclusively focusing on genetic mutations, which may be an effect rather than the cause of cancer.